Good morning to all of you. Today, I'm going to discuss about the topic that is application of biotechnology uh, under the chapter biotechnology and overview for class 11B biotechnology. So, what are different application of biotechnology in different fields? In the field of medical science, that is the health. In the field of agriculture, in the field of chemical industry, that is industrial application, then cleaning industry, then textile industry, and pulp and paper industry. So in previous class I have discussed the section that is the health industry, that is regarding the protein pharmaceutical production, then vaccine and therapeutic agent production through recombinant DNA technology, and then diagnostic kit production regarding preparation diagnostic kit like different disease detection kit, ELISA kit, and other pregnancy test kit, and so many kits are available in the market for diagnosis of different disease. Then gene therapy. So gene transfer technique, that is for treatment of defective gene that cause or genetic disease. So we can treat the defective gene or genetic disease through gene transfer technology. And you have learned the vaccine, DNA vaccine, recombinant vaccine, how they are going to produce through recombinant DNA technology. Then diagnostic kit like ELISA kit, how they are going to utilize for detection of different type of viruses. Then production of therapeutic agent like OKT3 repro. Repro is also used for treatment of cancer. That is the drugs that is produced through the recombinant DNA technology and biotechnology. Then vaccine or recombinant vaccine or DNA vaccine. Then protein, this production of different type of protein that can be used as enzyme, that can be used as antibiotics, that can be used as antibody like OKT3, that is used for kidney treatment, kidney transplantation. That is the therapeutic agents. Next, in agricultural industry, I have discussed the biofertilizer, like biofertilizer produced by Pseudomonas bacteria. The bacteria that is Pseudomonas, that is responsible for nitrogen fixation, that is blue green algae, right? That is cyanobacteria, and rhizobium bacteria. Then what is your role as a biotechnologist? Just improve the strain of that bacteria. Strain means variety of bacteria. Okay, so just they can do. They it can be used for increasing the efficiency for nitrogen fixation in the field, just to make the availability of nitrogen for the plants. Okay, biopesticide, that can be used for checking the growth of pest insect. Okay, then creation of transgenic plant. What is transgenic plant? You know, the plant that contain foreign DNA, and that due to which there is a improvement in the character, the feature, the quality of that plant regarding nutrient content like vitamin A, or protein content like any amino acid, or we can also concern with the creation of transgenic regarding the uh, biotic stress and abiotic stress. What do you mean of biotic stress? Biotic stress means that type of environmental condition that can damage the plant, that can affect the growth of the plant. Like, what is biotic stress? Attack of TD, attack of insect, attack of
Yes,
different
sir.
type of microbes like bacteria on the plants. So we are going to create a transgenic plant. What plant? Transgenic plant against that bacteria, that fungus, that virus, that insect. So cotton bollworm. Scientists have developed the BT cotton. You know, BT cotton, it is a transgenic crop. BT brinjal, it is a transgenic crop. Scientists have already developed that type of uh, variety of the cotton plants. So when there is attack of cotton bollworm, when there will be attack of cotton bollworm, there is no effect on the ball of cotton. So if they are attacking the juice sucked by the insect, it will kill that insect. How? You know, BT. BT is the gene, name of the gene. And BT gene that encode the BT crystal protein. BT gene, if they is present in a plant like cotton, like brinjal, it will encode the BT crystal protein. So this BT crystal protein generally soluble form in the plant liquid. But when the insect is sucking that liquid, when that juice of the plant is going to enter in the stomach of that insect, it become crystal, solid crystal, due to change of the pH, due to change of the pH of the environment inside the stomach of that um, insect. So that crystal form of BT crystal protein will rupture the cell membrane of intestine, also known as crop is the name of the intestine of insect. So thus it will kill the insect. So this is the advantage of using the transgenic crop. And it is also eco-friendly. How it is eco-friendly? Because farmer is not going to spray the insecticide, farmer is not going to insect the pesticide, because biopesticide means use of any living microbes that can check the other pest, other insect. So that is the concept. Means these are the biotech stress. Biotech stress means the attack of insect, attack of pest like microbes, attack of microbes, that is come under the biotech stress, attack of animals. So but if you are thinking about abiotic, means non-living component. What do you mean by non-living component? That is a scarcity of water. If plant is growing in a scarcity of water, then this is the good for farmer. There is no need of water. So such type of variety of the plant is required in the area where there is less rainfall. Generally, in some part of Rajasthan, there is scarcity of water. There is a scanty rainfall. So that situation, plant cannot grow. But if scientists have developed the variety that can grow even in a scarcity of water, that is drought resistant gene, DRG. DR means drought resistance. Okay. So if such type of gene is present in the plant, and if some osmolytes, chemical compound is synthesized by the plant itself just to overcome the problem of scarcity of water, in that situation also plant can survive. So there are so many chemicals are synthesized by plant itself, or if you can transfer the gene responsible for that chemical compound synthesis, if that chemical is synthesized by plant, then plant will able to tolerate the scarcity of water. Again, other factor like salt. If salt is present in the soil, plant will not grow, you know. So salt alkaline condition generally not favor the growth of the plant. Generally neutral pH of the soil that can favor the growth of the soil. But if your soil is alkaline, you know, pH more than 7 is alkaline, you will study these things in chemistry also. So such factor is affecting the growth of the plant. If the soil is alkaline, it means soil is not fertile, soil is barren. Okay. So in that situation, some plant is able to grow. How it is able to grow? If you transfer the gene to survive against that environmental condition, that type of soil condition. So that is the transgenic plant. So even in alkaline soil, we are able to grow the variety of plant, that is transgenic plant, against that alkali condition. That is the condition of uh, abiotic factor. Next abiotic factor is like abundance of water. If they offer water, over rainfall also affect the plant growth. So if a plant is submerged inside the water, there are two conditions. One is scarcity and one is overflow. Means plant is going to submerge inside water. you know this time is the time of a flood, and in the flood condition, if plant are submerged inside the water, it will not grow properly. It can be damaged by that water. So scientists have developed a variety of the rice. It can grow even in the 5-meter water. Plant is already submerged, and in that submerged water plant can grow. So that is the situation. Is crop tolerant, that is abiotic, means a scarcity of water as well as abundance, overflow of water, over excess rainfall of water. That is the come under the abiotic condition. Next is alkaline soil condition. It is also abiotic condition. So these are the different situation regarding the factor of abiotic that can affect the growth of the plant. So scientists have developed the plant against that condition situation, so that variety can be grow even in the area there is a scarcity of water, even in the area there is a over flooded condition. So that is the situation. Next, I already discussed the biotic condition like viral attack on the plant, bacterial attack on the plant, then fungal attack on the plant, then insect attack on the plant. Have you heard about the Tidda attack recently in Rajasthan, then Gujarat, then some area of Madhya Pradesh, some area of UP. So this is they are coming in the groups, and whole field is going to run by that insect. Means they are damaging the plants directly. They are eating the whole plants, leaves, food grain, and farmer cannot produce huge quantity of that food grain. In that situation, that is come in the biotic stress. So scientists have developed so many transgenic crop against that insect, that pest, that bacteria, that virus. Okay, so these are the concept. Next is pharmaceutical production. In the last class I have already discussed the molecular farming. Molecular farming regarding production of edible vaccine. Means you are going to create a transgenic plant, the gene responsible for antigen production. Suppose you are infected with hepatitis. So um, there is a protein present in the bacteria, virus. So that type of protein can be synthesized in the plant also, and the protein of the bacteria act as antigen. So if that antigen will enter in your body, it will cause disease. So small quantity of antigen that will trigger the antibody production. So that things can be produced inside the plant. So this is the concept of molecular farming, and production of useful protein or useful compound that used as pharmaceutical compound for treatment of different type of disease. So this is the concept of molecular farming. Molecular farming, that is the you can select the any commercial plant like wheat, like rice, like tomato, like sugar cane, like tobacco also. These are used for production of any chemical compound. Tobacco, tomato, then brinjal, tomato. So these are the model plants in which there is transfer of the gene that can use for production of antigen and antibody. Okay. So if you are eating that crop product, that like fruit, vegetable, then automatically you will cure from that type of disease, and this is the concept of edible vaccine. So all the concept will turn around technology that is known as recombinant DNA technology, then gene transfer technique through recombinant DNA technology, then plant tissue culture for multiplication of the plant. Okay, so the concept is just to isolate the gene from any organism, either from human body, from the animal, from the microbes, and transfer inside the plant just for production of any pharmaceutical compound. So that is the concept. CMAP, Central for Aromatic Plant Research Institute, that is present in the Lucknow, and Naipur is present in Mohali. Okay, so that is the area. That is the these are the institute where there is a production of pharmaceutical compound inside the plant. Okay, so this is concept. This concept is known as molecular farming. Naipur. Okay, and CMAP. CMAP is institute regarding production of aromatic and medicinal compound. That is there is so many research is going on different medicinal plant. Okay, so that compound is going to extract for treatment of different type of disease. Okay, that is known as secondary metabolite production. In the plant there is so many chemicals like secondary metabolite, but these plant are producing for protecting your that their cells, but human body are utilizing for their own purpose. What I am telling, if plant are producing the secondary metabolite or chemical vincristine, vinblastine in the flower, that is vinca rosea, and also lipstick compound that is produced by plant, lithospermum species. Okay, so these are the eco-friendly that can use for protecting for themselves only, but scientists have utilized. Okay, then so many parts of plant are producing such a secondary metabolite for protecting their self, but human are utilizing for treatment of different type of disease for themselves. Okay, so this is the concept for production of secondary metabolite, over production of secondary metabolite inside the plant. So scientists have developed a technique through genetic engineering and plant tissue culture, improve the function of the gene responsible for that secondary metabolite production, and that chemical extraction used for treatment of different type of disease. So this is the pharmaceutical production, and this process is going on mainly in the research institute, that is CMAP in the Lucknow and Naipur in the Mohali. So these are the institute in the CSIR institute. You know CSIR institute, Central for Industrial Research. Okay, यहाँ पे बहुत सारे कैरियर अपॉर्चुनिटी है, बहुत सारे आपको जॉब मिल सकते हैं CSIR में। अगर आप CSIR इंस्टीट्यूट से आपने कोई भी डिग्री ले लिया और कोई भी एक्सपीरियंस ले लिया, you will get lot of money after getting the degree from that CSR institute. आपके चंडीगढ़ में बहुत सारे CSR इंस्टीट्यूट है, एमटेक माइक्रोवेल इंस्टीट्यूट ऑफ टेक्नोलॉजी, और आपके पास में ही है 34-35 सेक्टर में CSR
Yes,
इंस्टीट्यूट,
sir.
ये पता है। नेक्स्ट है
सर
इस केम, हाँ बोलो।
सर मतलब वहां से डिग्री हो, कोई कॉलेज है CSIR?
CSR जो है एक ऑर्गेनाइजेशन है, ठीक है, वो अपना हर जगह ऑल ओवर इंडिया में इंस्टीट्यूट बना के रखा है, लैब बना के,
यस सर,
लेबोरेटरी
ये तो
वेयर
पता
देयर
है
इज
सर,
ए
हमारा कैंप भी
रिसर्च
लगाते हैं
वर्क
वहाँ
इज,
पे।
हाँ रिसर्च वर्क हो रहा है। तो मैं ये कह रहा हूँ कि जब आप बीटेक डिग्री लेंगे या बीएससी डिग्री, जैसे ही एमएससी डिग्री लिए या एमटेक लेंगे, तो आप एक एग्जाम कंप्लीट करेंगे, नेशनल एलिजिबिलिटी टेस्ट। इसमें जैसे ही आप क्वालिफाई कर जाएंगे, तो ड्यूरिंग स्कॉलरशिप मिनिमम इंडिया में मैं बात कर रहा हूँ 28,000 दिया जाता है, ओनली फॉर स्टडी पर्पस। और एचआरए आप समझ लो, उसका एचआरए तो लगभग 32-35 के आसपास मिलेगा ड्यूरिंग स्टडी पीरियड व्हेन यू आर टेकिंग द डिग्री। ओके, so when you are doing the degree like BTech, and MTech, MSc, then you have to qualify that exam. और जो कुछ मैं बता रहा हूँ, जो कुछ भी पढ़ा रहा हूँ, ये सारे कुछ पूछा जाएगा एज ए बायोटेक्नोलॉजी और लाइफ साइंस. And some portion will question will ask from the for the GK in from physics also, chemistry also, some portion, not all. Okay, so major question will ask from biotechnology. Okay, so you will get benefited when you will qualify. मैं भी क्वालिफाई किया हूँ ये सारे जो नेट होता है, इतना बार, 2 बार. So there are different type of eligibility test that is going on in India. कौन कौन सा? एक है CSIR, सेंट्रल फॉर इंडस्ट्रियल रिसर्च इंस्टीट्यूट. दूसरा है
CSIO.
ICAR, इंडियन एग्रीकल्चर रिसर्च इंस्टीट्यूट, ICAR. इसका भी नेट एग्जाम होता है, ये भी फेलोशिप देता है जस्ट लाइक CSIR. एक होता है ICMR, इंडियन काउंसिल ऑफ मेडिकल रिसर्च. जो कुछ भी वैक्सीन बनेगा तो ICMR इज कंट्रोलिंग द ऑल द मेडिकल इंस्टीट्यूट, मेडिकल कॉलेज, मेडिकल लैब. ओके, मेडिकल रिसर्च ओरिएंटेड लैब. जो कुछ भी आपका ऑथेंटिसिटी मिलेगा, ये कौन करेगा? ICMR. सो ICMR का हेडक्वार्टर दिल्ली में है, दैट इज अटैच विद एम्स. ओके, तो ICMR का मुख्यालय दिल्ली में है, CSIR का मुख्यालय भी दिल्ली में है सारे का, और ICAR का भी मुख्यालय जो है हेडक्वार्टर दिल्ली में है. So that is a controlling body, and they are funding the scholarship and the research project just to develop the different type of variety, different type of strain of bacteria, different type of medicine. Okay, NIPER, this is also a national institute, a microbial institute. अगर आपने कैसे भी इंटर कर गया उस माइक्रोबियल इंस्टीट्यूट में तो पैसे की कमी नहीं है. 2 लाख 3 लाख पर मंथ तो तुरंत आएगा. फिर आपको प्रोजेक्ट हैंडल करोगे करोड़ों का. So when you will start the dealing the project, means you will working with the project. So when you will gain the experience, gradually your income will increase. Okay, so there are lot of opportunity in the field of biotechnology. और किसी भी CSIR इंस्टीट्यूट है, CDRI, सेंट्रल ड्रग रिसर्च इंस्टीट्यूट, लखनऊ में है. ठीक है ना. CMAP ये लखनऊ में है. ठीक है ना. इंटरनेशनल सेंटर फॉर जेनेटिक इंजीनियरिंग, इंटरनेशनल सेंटर फॉर जेनेटिक इंजीनियरिंग ये दिल्ली में है. ये मैं कह रहा हूँ, ये जो जहां जहां मैं बोल रहा हूँ, अगर उसमें आप कैसे भी एंट्री कर ली अच्छे से पढ़ाई कर, फिर पीछे मुड़ के नहीं देखना है. there is lot of job opportunity, lot of scope for research, and you will get
सर
yes.
इनके लिए करना क्या पड़ता है, मतलब कौन से पेपर करने पड़ते हैं, कौन सी
मैं ये कह
स्ट्रीम
रहा हूँ
चूज
कि
करनी
उसके लिए
पड़ती है?
आपको बायोलॉजी बायोटेक्नोलॉजी पढ़ना है. बायोटेक्नोलॉजी मेन पढ़ना है, बायोलॉजी तो आप पढ़ते ही रहते हो. मैं ये कह रहा हूँ कि बायोटेक्नोलॉजी पढ़ना है और केमिस्ट्री पढ़ना है. केमिकल इंजीनियरिंग में भी
नहीं
बायोटेक्नोलॉजी
सर,
है. ठीक है ना.
सर जैसे हमने +2 कर ली.
+2 कर लिए तो आप देंगे ना इंजीनियरिंग का एग्जाम, इंजीनियरिंग का एग्जाम दोगे ना.
यस सर.
तो जैसे
यस
इंजीनियरिंग
सर.
का एग्जाम देंगे तो आपका रैंक आएगा. मान लो आपको मन कर रहा है बायोटेक्नोलॉजी में जाना, तो आप बायोटेक्नोलॉजी में कहीं भी ऑल इंडिया में कहीं भी ले सकते हैं. चाहे आप IIT में ले सकते हैं, ये NIT में ले सकते हैं एडमिशन और बीटेक कोर्स. ओके, तो IIT में, IIT
यस.
खड़गपुर में है, IIT रुड़की में है, सब जगह है. मैं IIT दिल्ली में है, सारे जगह. IIT मुंबई में है. बायोटेक्नोलॉजी इज सेपरेट ब्रांच. Okay, so if you visit the lab, then you will understand what is biotech. आप तो अगर आप नहीं गए हो लैब में तो एक बार जाएंगे, अगर मौका मिलेगा तो सबको मुझे ले जाना बायोटेक्नोलॉजी लैब में. कहाँ कहाँ? चंडीगढ़ में सारे मैक्सिमम लैब में घुमाना. 2 लैब हमारे मेन है. एक माइक्रोबियल इंस्टीट्यूट ऑफ टेक्नोलॉजी, दूसरा है आपका ये जो 34 या 35 में है CSIR लैब, जहाँ पे बच्चे जाते रहते हैं. तो वहाँ पे यू कैन
अभी मैं
ऑब्जर्व
हूँ.
व्हाट इज द योर कैरियर in of future. In future, what is your career in the field of biotechnology? Means you will find lot of scope. तो अलग अलग डिवीजन है बायोटेक्नोलॉजी में भी. फर्मेंटेशन टेक्नोलॉजी अलग है, बायो प्रोसेसिंग अलग है, फिर रिकम्बिनेंट डीएनए टेक्नोलॉजी, प्लांट टिश्यू कल्चर अलग है, मेडिकल बायोटेक्नोलॉजी अलग है। मीन्स यू विल एबल टू डू द रिसर्च। कोई भी जाता है एमबीबीएस करके भी जा सकता है, ठीक है, या नहीं तो प्लेन आप जनरली बीटेक कर लिया और आप फिर रिसर्च के लिए आप वहां चले गए। तो गवर्नमेंट आर फंडिंग टू मच अमाउंट, sufficient amount of money in India, not only in India. Maximum funding is going on by USA, Germany, Japan. Japan are providing largest amount of scholarship. Means all over world में maximum amount money is paid by Japan. लेकिन प्रॉब्लम क्या है, वहाँ के खाने पीने का स्टाइल अलग है, जस्ट लाइक चाइना, ठीक है ना। तो बहुत
तो
सारे
हम
बच्चे,
चेंज कर लेंगे सर।
हाँ, तो मैं
सर
ये कह
हमारी
रहा हूँ
क्लास
कि
में है भी चाइनीज लड़का है।
हाँ हाँ, तो चाइना में भी है, लेकिन मैं ये कह रहा हूँ कि जापान में जो फंड देगा आपको, वो फंड आपको ऑल ओवर इंडिया वर्ल्ड में कहीं नहीं मिलेगा। यूएसए में भी देता है, मेरा फ्रेंड एक यूएसए में प्रोफेसर है, ठीक है। तो मैं ये कह रहा हूँ कि ऐसा नहीं है कि आप यहाँ इंडिया के हो तो आपको नहीं मिलेगा। यू विल गेट लॉट ऑफ फंड फॉर डूइंग रिसर्च, फॉर डीलिंग द प्रोजेक्ट। तो पैसे की तो बात ही सोचना ही नहीं है, and then you will get a name and fame also. Suppose you are doing some uh, uh, different research, novel research, then you will get opportunity. So this is the concept of biotechnology. And not, this is not only in foreign only, India also lot of institute are there in all over the country. uh, that lab is running in all over the state, okay. And you can do that research in a better way if you will qualify for CSR net or ICR net or ICMR net, any net. So you will get the fellowship and you will join any project under any scientist, okay. And then gradually you will get the degree, like MTech degree or MSc PhD integrated degree, like that. So many of course option are there. तो मैं ये कहना चाह रहा हूँ कि पेपर एंड पल्प दिख रहा है आपको, पेपर एंड पल्प इंडस्ट्री, so that is also come under the biotechnology. Means you are going to develop such type of plant that is going to produce that type of wood that is utilized for paper. So you are going to improve the quality of that wood, okay, quality of that leaf of that plant that is going to utilize for making paper. So paper and pulp industry, there are so many aspects. Bio bleaching of the pulp means you are going to remove the colors of the leaves material that is used for making the paper. Means you are going to remove the color of the leaf of plants just through recombinant DNA technology or through biotechnology. So you will get the opportunity in paper and pulp industry, that is a separate branch in Roorkee, IIT Roorkee, okay. And improvement of physical property of fiber also. You can improve the quality of fiber of the cotton, quality of fiber of the jute, quality of fiber of the silk. So silk, on the silk, so many research is going on in CCMB, Central. Okay, so CCMB, it is laboratory, CSR laboratory is present in the Hyderabad Center for Cellular and Molecular Biology, CCMB. Okay, and Center for DNA fingerprinting also present in Hyderabad, CFDR. Okay, so then Center for Forensic Research also present here in चंडीगढ़। फोरेंसिक रिसर्च जो कि इन्वेस्टिगेशन करता है ना, पता है ना किसी भी क्राइम में फोरेंसिक साइंस का कितना रोल है। कोई भी किसी का मर्डर होता है तो उसमें एक हेयर से उसमें से डीएनए निकाल के पता लगाया जाता है किसका डीएनए, सस्पेक्ट किया जाता है, ठीक है ना, पता है ना। एक स्पॉट, one drop of spot of the your blood that can use for detection of the person, ठीक है ना। किसी का ब्लड गिर गया वहां पे एट द टाइम ऑफ किलिंग द पर्सन, और किसी का हेयर गिर गया, हेयर गिरने से भी हो गया, और किसी जिसको कोई मार रहा है उसने नेल से उसको नोच लिया तो उसके साथ उसका मसल आ गया, तो दैट मसल्स कैन बी यूज्ड फॉर एम्प्लीफिकेशन ऑफ डीएनए, and through that muscle DNA can be amplified and can detect that person. So that is the forensic science. तो इस तरह से आप कर सकते हैं, एनी फील्ड यू कैन डू इम्प्रूव योर रिसर्च। देन केमिकल इंडस्ट्री मैं बता चुका हूँ कि फर्मेंटेशन प्रोसेस टू प्रोड्यूस ऑर्गेनिक केमिकल in presence of oxygen with the help of microbes that is going to produce any useful compound like use of pseudomonas, use of lactobacillus. These are so many yeast, use of yeast that is used for production of alcohol, acetic acid, aspergillus that is the fungus. फंगस का यूज करके आप विटामिन सी प्रोड्यूस कर सकते हैं, एसिटिक एसिड प्रोड्यूस कर सकते हैं, बैक्टीरिया को यूज करके प्रोपाइनोबैक्टीरियम बैक्टीरिया उससे आप विटामिन B12 बना सकते हैं। तो न्यूट्रिशन रिसर्च इंस्टीट्यूट दैट इज प्रेजेंट also in Mysore. Then you can also do the research in the food science through food biotechnology. Okay, so that is CSR lab present in Mysore, very nice place, just like Chandigarh. So that is the place where you can do the research and you gain the experience. Okay, so utilization of so many bacteria, fungus, virus that is used for production of different type of chemicals, mainly bacteria and fungus. Okay, so yeast is a fungus used for production of alcohol. Propionobacterium is a bacteria that is used for production of vitamin B12. Then you know other aspergillus niger. Aspergillus is the fungus that is used for production of citric acid or acetic acid. So production of high purity chemicals also produced inside the chemical industry, and usage of energy efficient process means you are going to process the raw material. What is the raw material? It can be a straw of the plant product of any cereals that can be converted into useful chemical compound. That is the application part of biotechnology, means you are going to improve the same amount of straw have less value, but same amount of chemical that has more value in the market. This is the concept. Means what is the utilization of bacteria and fungus that going to convert the raw material like a straw of the plant part after harvesting, after threshing, you are going to produce more useful compound, that is bioprocessing engineering, that is chemical inside the chemical industry with the help of microbes. We are going to produce useful chemical, that is alcohol, butanol, acetic acid. So these are the chemical. Use of energy efficient process. This you are going to use the uh, uh, algae. You are going to use the straw for producing the energy compound, biofuel, जेट्रोपा प्लांट। जेट्रोपा प्लांट से आप बायोडीजल बना सकते हैं, ठीक है ना, उसके लीफ से, उसके फ्रूट से। सो दैट इज द कंसेप्ट। एंड ऑल्सो यू कैन यूज द प्रोड्यूस द बायोडीजल फ्रॉम एल्गी। एल्गी से भी बनता है बायोडीजल, आपको समझना है। तो अगर आप किसी न किसी एरिया में अगर अच्छा से रिसर्च करेंगे तो पैसे की कमी नहीं है। देयर इज लॉट ऑफ मनी, लॉट ऑफ स्कोप इन द रिसर्च। देन क्लीनिंग इंडस्ट्री, आपको बताया था ना, क्लीनिंग डिटर्जेंट लाइक प्रोटीएज, सबटिलिस, सबटिलिसिन। क्लीनिंग इंडस्ट्री में समझ में आया कि ये जो सबटिलिसिन है ये एंजाइम है, प्रोटीएज ग्रुप का है। अब उस सबटिलिसिन जो कि बैक्टीरिया से निकलता है, कौन सा बैक्टीरिया? बैसिलस सबटिलिस। फ्रॉम द बैसिलस सबटिलिस these are going to produce the enzyme that is सबटिलिसिन। लेकिन ओरिजिनल जो स्ट्रेन बैक्टीरिया है वो सॉइल में होता है। और अगर दैट सबटिलिसिन इज गोइंग टू यूज फॉर क्लीनिंग, that is mixing with the detergent, there is no improvement in cleaning action. But when you are do the side directed mutagenesis, this is the technique of biotechnology, then you are going to improve the quality of that bacteria at the gene level. So you are going to change the gene, क्या gene? Gene of methionine, एमाइनो एसिड होता है। ये आप पढ़ेंगे अभी, क्या अभी बताएंगे बायोमोलिक्यूल्स में। कार्बोहाइड्रेट है, प्रोटीन है, फैट है, विटामिन है। तो प्रोटीन का जो स्मॉलेस्ट यूनिट है अमाइनो एसिड, उसमें मिथ्योनिन होता है 222 पोजीशन पे, जिसको आप चेंज कर देते हैं जीन लेवल पे। तो ये सिस्टीन में चेंज होता है या एलानिन में, जिसके चलते इसका जो क्लीनिंग एक्शन प्रॉपर्टी इंप्रूव हो, क्या हाई क्वालिटी क्लीनिंग करने लगेगा। कौन सा? द सेम डिटर्जेंट दैट इज मिक्स विथ इम्प्रूव स्ट्रेन प्रोडक्ट ऑफ सब्टिलिसिन, जो कि इम्प्रूव स्ट्रेन से बना है सब्टिलिसिन। So this is the advantage of cleaning industry. Means there is lot of scope in cleaning industry for biotechnologists. Next is textile industry. Use of enzyme for finishing of fabric. Means you can produce so many different type of enzyme that is used for finishing the fabric. Means improve the quality of fabric, means cloth. You know fabric, a fiber that is the unit of fabric, and fabric that is after weaving process we get the cloth that is known as fabric, आपको पता है। तो ये फैब्रिक को का क्वालिटी इंप्रूव करना है कि सॉफ्टनेस बढ़ाना है, कलर चेंज करना है, तो दैट इज द कंसेप्ट दैट विद द यूज ऑफ एंजाइम। देन यूज ऑफ जेनेटिक मॉडिफाइड कॉटन दैट इज नोन एज जीएमओ कॉटन, जीएम जेनेटिक मॉडिफाइड कॉटन दैट इज नोन एज बीटी कॉटन। So BT cotton that is that have more important because they are giving the advantage to the farmer. Not only the farmer, the textile industrialist also, so that they know effect of the uh, insect like cotton bollworm, and that not damaging the fiber. So that cotton bollworm that will damage the ball. You know ball is the fruit of cotton. In class 7 you have a study. Ball, what is ball? Ball is the fruit of the cotton, and inside the ball there is a fiber. What happens when the cotton bollworm attack? It will degrade whole ball. What happens? Whole ball will destroy of any field. Then what farmer will you get? They have applied the fertilizer, they have applied the seed, they have purchased the seed, they have purchased the fertilizer, they have purchased the insecticide. So what they will get? If farmer is selecting the BT cotton seed and growing that seed, then there will be no attack of that cotton bollworm insect, and farmer will get benefited. Okay, so they will get maximum return in the form of money after selling the fiber, cotton fiber, and this will also advantages for industrialists also for getting good quality of cotton. Next is the pulp and paper industry. Improve the physical property of fiber. You know, fiber that is used for making what? Making of paper. So best quality of paper, for making best quality of paper that is going to utilize for making the books, making the currency. That is through recombinant DNA technology and plant tissue culture technique, and then use the paper and pulp techniques. So that is the separate branch in the field of biotechnology, that is paper and pulp industry, where biobleaching of pulp, that is the materials that is used, uh, semi-liquid material that is used for making the paper. So that is going to improve. We have to remove the color of the greenish color or yellowish color or brownish color of plants, so that we get the good quality of paper. That is the white quality of paper. So such area we have lot of scope just for job point of view, for earning point of view, and give the job point of view to others also. So you can give the job also after establishing your own industry in the field of plant tissue culture, in the field of paper and pulp industry, in the field of chemical industry, in the field of cleaning industry, that is the detergent industry. So you can also establish your own business in the field of biotechnology by choosing any area. So this is the concept. Next is what is advancement of 21st century has witnessed this. क्या क्या हुआ है अभी तक बायोटेक्नोलॉजी में? तो मैंने बार बार बोला है कि क्लोनिंग एक्सपेरिमेंट हुआ है, लिख रहा है ना इसमें, क्लोनिंग ऑफ एनिमल किसका हुआ था? सिप का हुआ था, डॉली बना था, और डॉली में फिर से उसका बेबी बना। तो ये जो एक्सपेरिमेंट ऑलरेडी किया जा चुका है। मैपिंग ऑफ जीनोम, मतलब जीनोम का सीक्वेंसिंग किया जा चुका है, ह्यूमन जीनोम का भी सीक्वेंसिंग किया जा चुका है। ह्यूमन जीनोम का सीक्वेंसिंग सबसे पहले वाटसन एंड क्रिक मॉडल किया दिया था ना, डबल हेलिक्स मॉडल। तो किसका किया गया था सबसे पहले? वाटसन का, साइंटिस्ट वाटसन था। और ह्यूमन जीनोम प्रोजेक्ट 2003 में ही कंप्लीट हो चुका था, ठीक है ना। तो इस तरह से ये जो है एनिमल स्पेसीज इंक्लूडिंग ह्यूमन आल्सो। देन डेवलपमेंट ऑफ टारगेट स्पेसिफिक न्यू molecule and drugs, you can there is a lot of opportunity. So many organization is going on for development of new type of drug. Okay, stem cell therapy for genetic disease and accident case. Okay, accident case में जैसे कॉर्निया लॉस्ट हो गया, तो एनिमल सेल कल्चर से आप डेवलप कर सकते हो कॉर्निया। देन जेनेटिक डिजीज को ट्रीटमेंट करने के लिए आप जीन थेरेपी करते हैं। टिश्यू इंजीनियरिंग फॉर प्रोड्यूसिंग आर्टिफिशियल टिश्यू एंड ऑर्गन, that is the from your cell we can develop your whole organs, like your limbs, like your heart, like your kidney. So like that is the development of particular organs. Okay, limbs means hand and legs. Okay, and your finger. Then creation of transgenic animal for production of recombinant bio pharmaceuticals, means molecular farming. Not only in plant, we can also use the animal for the production of any chemical compound. And from the fish, from the meat, from the milk, we get that chemical compound. So we create a transgenic animals and transgenic plant. That is the concept of molecular farming, PHARMING, and other is molecular FARMING, molecular farming. Molecular farming is for plant, just to develop the edible vaccine, a concept, production of any antibody, production of any यूजफुल कंपाउंड दैट इज यूज फॉर ट्रीटमेंट ऑफ डिफरेंट टाइप ऑफ डिजीज। तो ये सारे कंसेप्ट है। इमरजेंस ऑफ बायो इन्फॉर्मेटिक्स एंड कंप्यूटेशनल बायोलॉजी, तो ये मैं कहना चाह रहा हूँ कंप्यूटेशनल बायोलॉजी दैट इज द एप्लीकेशन ऑफ द कंप्यूटर साइंस इन द फील्ड ऑफ मॉलिक्यूलर बायोलॉजी टू अंडरस्टैंड द फंक्शन ऑफ एनी बायोमोलिक्यूल एट molecular level, to develop a different software. Okay, so different software are available in the field of genomics, in the field of proteomics, just to understand the function of the gene, understand the function of protein. We can develop a different model of the gene, DNA, protein for drug design. Okay, so application of in silico experiment to understand the pharmacology, toxicology. आपने थोड़ा सा भी डिग्री ले लिया इसमें फार्मेकोलॉजी में, फार्माकोलॉजी इंस्टीट्यूट है लखनऊ में ऑलरेडी, तो आपको जॉब मिलना ही मिलना है। टॉक्सिसिटी में आपने थोड़ा भी डिग्री ले लिया, तो दैट कैन यूज फॉर टेस्टिंग एनी केमिकल दैट हैव हाउ मच टॉक्सिसिटी देयर, दैट कैन अफेक्ट योर हेल्थ. So then development of automated gene sequencing, protein sequencing of different organism, then identification of protein